Online people, 6.2, page 251. Something called inheritance of linked genes. Period. The lab's not due. I don't know. I can tell you, like, and I'll have a better understanding once I finish the lesson on linked genes, and then I kind of like gonna talk to you and monitor progress. I have no idea. All right. So we talked about, like, I know we're all familiar with the law of segregation. Can anyone remind me of what segregation is? Segregation during class starts at, I believe, 1:10. 1:10? Okay, 1:10 on Fridays. All right. Can you please remind me of what segregation is, or the law of segregation is? Page 251, section 6.2. And you probably won't be able to just recollect law of segregation looking at the diagram, looking at 251. So in meiosis, when you have the homologous chromosomes to get pulled to two different cells, you remember that situation? You don't remember this situation because all of you have a blank look. So all right, this is a simple enough diagram. We're going to take a look at this and try to recall things. In meiosis, let's start with the diploid cell, right? And you have two, your chromosomes are in two different colors, meaning, say, let's assume the purple ones came from the mother and the blue ones came from the father. Then you have a set of chromosomes, right? A complete set of chromosomes that need to, or that will separate. Does that make sense? How many chromosomes do you see here? Four. So by the time meiosis happens, how many chromosomes are you going to get in each cell, in each daughter cell? A Hassan or a student? It's cool. Why are you laughing at me? I practice my Arabic with them. And he's beginning to understand my horrible Arabic. So yesterday he threw away a mouse. Right? There was a baby mouse dead outside. No, there was like an actual mouse. So I saw it. I'm like, I'm Hassan, far. And he comes, he goes, Khalid. I'm like, no, you gotta move it, right? And he doesn't. So I come in, and then I changed my mind about the mouse. I want to get it. So I took the grade 12 out. I'm like, where's the mouse? And he goes, khalas. I'm like, no, you cannot khalas it. You just like that, you just had it there. So yeah, so it's Arabic. He's beginning to understand my Arabic, and then yeah, it's kind of fun. Anyway, so take a look here. So you have a diploid cell. Then when they separate, right, the chromosomes will independently assort regardless of what their origin was. So in the new daughter cell, you can see it's a mixture of both mother and father chromosome, right? It's not that if you're going to make two daughter cells, one of them will get only the mother chromosome, the other one will only get the father chromosome. Does that make sense? Does that make sense? And it's no rule like that, whether you're going to get, like, we have 46, the 23 one, 23 other one, or 10 of one. One cell gets 10 of the mother, and then another one gets like whatever of the other, right? So if they independently and randomly assort, what does it do? In terms of diversity, what does it do? Genetic diversity, increase or decrease? Increases it. Before that, what happens even before the law of segregation or before the segregation happens, something else happens. Crossing over, and crossing over does something else. What does it do? Yeah, so it exchanges genes. So you see that the blue one is like all from the father, but they will have now a section of mother's genes as well, right? So we, as long as you're clear on that, we can go on. So law of segregation, law of independent assortment. Siblings for genes segregate independently of their origin during meiosis, and that's kind of cool. But sometimes, if the genes are on the same chromosome, like for example, there's a hair color gene, and then right underneath there's the eye color gene. If they're going in crossover, both of them are going to cross over. Does that make sense? If not, I'll repeat it one more time. Okay. So if, for example, in this chromosome, let's see, mother's chromosome, father's chromosome, right? If this is a gene for, say, insulin production, the other one is a gene, this locus, there's a gene for body mass ratio, right? If this section is going to get exchanged with this section, both the genes are gonna go together. It's not gonna, you're not only gonna get the insulin gene from the mother, you're also going to get the body mass index gene that goes with it. It's like you get one, you get the other. Does that make sense? All right. Let me use more concrete examples for this. Check over here, and I want you to kind of recall back to Mendelian genetics. So I have purple flower, and then you have red flower, okay? Which trait, oh, sorry, we're looking at dihybrid cross because we're looking at two different traits, color and pollen shape, okay? Which one is the dominant phenotype or the dominant genotype? Because the red and round disappear from the first generation, F1 generation, yes? Online, everyone clear? Okay. We know that Mendelian genetics, if you were to cross the F1 heterozygous, F2 would have a very particular ratio. Do you want to? No, this is dihybrid 9. Yeah, so 9 to 3 to 3 to 1. But in this case, that ratio changes. You see that? Because what happens is the purple tends to, or the long pollen grain tends to be inherited with the purple flower, and the round pollen tends to be inherited with the red color. So somehow the linked genes are the genes for color and pollen shape. Okay, so certain genes just tend to, they just happen to get inherited together. You get one, you're gonna get the other one. It doesn't mean that you're not gonna get some of the mixed match. You're gonna get that, but a smaller ratio, much smaller ratio would get that sort of a mixture. Does that make sense? Okay, and you can tell that. It's the easy way to tell if you can't see the Mendelian ratios anymore. Make sense? Okay, so after this, you should actually be able to finish the rest of your lab. You ready for that? Okay, if you don't understand this, let me know. I'll go over it again. Okay, tell me where to start. Tell me where to start, and I will start there again. This one? This one? Okay. So I have, can you give me how many traits am I looking at? I'm looking at 2 traits. Therefore, I'm looking at a dihybrid cross. Agreed? Okay. I have a dominant trait. Yes, my dominant trait is purple color and long pollen grains. Cool. I can also make an assumption or conviction that purple is the dominant color and the long pollen grain is the dominant shape. Yeah, because I don't see that anymore. Okay. We know from Mendelian genetics or the ratio that if, is this heterozygous F1 generation, is this heterozygous for both the traits? Yeah, because it is a combination of these two, heterozygous. If you were to self-pollinate F1, then you should have technically had a 9:3 ratio, meaning 9 of them should have had purple and long. 3 of them should have been purple and round. 3 of them should have been red and long, and then 1 of them would have been red and round. Only recessive traits, 1. Okay, but I don't see that anymore. Do you see what you got? What do you get?
One of each trait.
Yeah, so you get, you don't get that ratio anymore. What's the ratio here? The majority of them are going to be, check the numbers there up there.
Yeah, 15.6.
15, let's say 16 to 1 to 1 to 5. Okay, so most of them tend to be purple and long. Yeah, why? Because if something during crossover is getting crossed over, they tend to get the color gene for purple as well as the long pollen grains together. It's inherited together. Okay, if you get that, you also get that. All right, so that's that. That should be, so if you see that kind of a frequency, you can assume that those genes tend to go together. And the reason that would happen is because they're close together on the chromosome, okay? If they're far apart, that will not happen. We can go to the gym and finish our work. Thank you. No difference in that one. Yes, you're right. So in F1 generation, you're not gonna see difference, but F2, yes. The F2 would have unusually high ratio for both the dominant traits. Yeah. All right, do you need me to come upstairs with you guys, or are you good? Just your room is usually upstairs, right? Thank you. All right, we gotta go because we do have the lab to finish. And when I get working, finish at 5 onwards, and then I'll help you with the question that you have section 4. Go, you'll run out of time.
Just in case I'm not in the lab, I have a dentist appointment and I was gonna come on 15,
When?
like 2:20.
2:20?
Yeah, around that.
Oh,
time.
but you're there now.
Yeah, I'm gonna
It's
be
only
there.
1:35, so you have plenty.
Yeah, just
It's
saying,
okay,
just in case.
yeah, don't worry.
But,
Welcome, son.